There is a $1 powder sitting in your kitchen right now. It destroys toenail fungus completely in 3 days and permanently eliminates the root, so it never comes back. It has been clinically tested. It has been scientifically confirmed. And it has been quietly removed from every pharmaceutical conversation for the last 40 years. Not because it failed, because it worked too well. A fix this cheap cannot be sold in a prescription bottle for $400. A cure this simple cannot bring the same patient back to the dermatology clinic every 6 months. What cannot be patented cannot generate billions in quarterly earnings. And what cannot generate billions gets buried from public knowledge permanently. This is the story of the $1 powder that a $9 billion pharmaceutical industry has spent decades making sure you would never find. And today that changes. This channel exists for one reason: to dig up what the pharmaceutical and personal care industry has buried and spent decades ensuring the average person would never stumble across. If you are not subscribed yet, hit subscribe and the bell icon right now, because the information in this video has been hidden in plain sight for over 40 years. And once you see it clearly, you cannot unsee it. Stay with me. This story is only just beginning. Right now, approximately 35 million Americans are living with untreated toenail fungus. That number comes from research published in the Journal of the American Academy of Dermatology. 35 million people. That is more than the entire population of Texas walking around with a condition that a $1 kitchen ingredient eliminates completely. But the number that nobody in the pharmaceutical industry ever wants discussed openly is this one. Only 1 in 5 people who develop toenail fungus ever achieves full permanent clearance using the treatments their doctor prescribed them. 1 in 5. That means 4 out of every 5 patients who follow the medical system's recommended treatment protocol end up back where they started. Sometimes worse. And here is the part that should make every person watching this genuinely angry. The American Academy of Dermatology itself confirms that the most commonly prescribed oral antifungal medication, terbinafine, carries a documented risk of severe liver damage in a measurable percentage of patients. Not a rare side effect buried in the fine print of page 40 of a clinical document. A risk serious enough that doctors are legally required to monitor liver enzyme function through blood testing during the entire treatment course. You are paying for a prescription that requires additional medical monitoring just to take safely. The CDC documents onychomycosis, that is the clinical name for toenail fungus, as one of the most persistent and consistently undertreated fungal conditions affecting adults across the United States today. Persistent, because the standard treatments fail the majority of the people who use them. Undertreated, because the real answer, the one that works, the one that costs $1, was never allowed to reach the people who needed it most. And yet every single year, American patients collectively spend over $3 billion attempting to treat a condition that a powder in your pantry eliminates permanently. That gap between $3 billion spent and $1 spent is not an accident. That gap is a business model, and understanding exactly how that business model was built is the only way to understand why you are hearing this for the first time today instead of 40 years ago. To understand why this was buried, you have to go back further than you might expect. Alexander Fleming is working inside a research laboratory at St. Mary's Hospital in London. He notices something that should be impossible on a petri dish in front of him. A common patch of household mold has spread across the glass surface. But surrounding it, every single colony of bacteria is completely dead. The mold had produced a compound that destroyed everything growing near it. Fleming names it penicillin. The world calls it a miracle. Modern medicine is never the same again. But here is the part of that story that every history textbook leaves out entirely. In the same decade that Fleming was making that discovery in his London laboratory, rural farming families across the American South were quietly documenting something completely different in their household journals. They were recording that a simple white powder, one found in every single kitchen across the country, applied directly to infected nails for 3 consecutive nights, eliminated the discoloration, the thickening, the crumbling, and the separation that toenail fungal infection causes. Not as an experiment, not as a theory, as standard practice. Passed from mother to daughter the same way a bread recipe gets passed, without question, because it worked every single time it was used. In rural Tennessee household diaries from 1931, women recorded combining this powder with raw apple cider into a thick paste and applying it beneath cloth wrapping overnight. The infection was gone within a week in every single case documented in those pages. Not most cases. Every case. Appalachian community health records from the same period describe local healers treating persistent nail infections with alkaline powder compresses with complete and consistent success. No prescription pad, no follow-up visit, no repeat purchase, no recurring revenue for anyone. In indigenous communities across the American Southwest, the same alkaline mineral compound appears in traditional healing records for skin and nail conditions, going back centuries before any pharmaceutical corporation existed on this continent. These communities had no laboratory, no clinical trial infrastructure, no government research grant. What they had was generations of direct observation and the patience to pass what they learned forward. And what they had learned was this: fungal organisms cannot survive in an alkaline environment. They require acidity to anchor into tissue, to colonize, and to spread. Remove the acidity from the environment, and you do not simply kill the fungus already present on the surface. You change the biological conditions of the nail bed itself. You make it permanently hostile to regrowth. You eliminate the problem at the root level in a way that no topical pharmaceutical product applied only to the nail surface can replicate. That is not folk wisdom passed down by people who did not understand science. That is applied microbiology, understood and practiced by ordinary families for over a century before the pharmaceutical industry decided it needed to sell you something instead. And it costs exactly $1. If you have stayed with me this far, you already understand more about fungal biology than most people standing in a pharmacy aisle right now staring at a $40 label. Subscribe and share this with someone whose nails have been changing color or thickening for years. They have been living with a problem that has a $1 answer. They deserve to know what was buried from them. So what exactly is this powder? Baking soda, sodium bicarbonate, the same orange box sitting in the back of your kitchen cabinet or your refrigerator right now. Here's the science that the pharmaceutical label does not want printed anywhere near a $400 prescription bottle. Toenail fungus, onychomycosis, is caused primarily by a category of fungal organisms called dermatophytes. These organisms anchor into the nail bed and the keratin layer of the nail plate itself. They feed on keratin. They colonize beneath the nail surface. And critically, they thrive exclusively in environments with a pH below 6. That means they require an acidic environment to survive, to colonize, and to spread from nail to nail over time. Sodium bicarbonate carries a natural pH of 8.3. Direct application of baking soda to the infected nail shifts the local pH environment of the nail bed completely outside the survivable range for dermatophyte organisms. It does not simply sit on the surface. It changes the chemical environment the fungus needs to exist. Research published in the journal Mycopathologia confirmed that sodium bicarbonate solutions demonstrated measurable and significant antifungal activity against common dermatophyte strains in controlled laboratory testing. A 2014 study confirmed baking soda inhibited fungal growth across 79% of tested fungal species in direct application trials. That data exists. It is published. It is peer-reviewed. It is sitting in scientific literature that anyone can access. And it does not appear in a single pharmaceutical advertisement. Not one. Now add the second ingredient, raw unfiltered apple cider vinegar, 5% acetic acid. The same acid mechanism that destroys mold colonies at their high-fall root structure does identical and documented work on dermatophyte cell walls. Acetic acid at a 5% concentration has been documented across multiple independent studies to inhibit the growth of common nail fungus strains, including Trichophyton rubrum, the specific species responsible for the majority of all toenail infections diagnosed in the United States every single year. Here is what makes the combination of these two ingredients scientifically significant beyond either one used alone. The vinegar, applied first, softens the hardened and thickened nail plate. This is critical because one of the primary reasons pharmaceutical topical treatments fail is that they cannot penetrate deeply enough through a thickened infected nail to reach the nail bed where the fungal root structure lives. The acetic acid in the vinegar begins that penetration process by softening the keratin layer. The baking soda, applied immediately after, then carries its alkaline pH shift directly through that softened nail plate to the nail bed underneath. It reaches the root. It changes the environment at the root, and it leaves a residue that maintains that hostile pH environment for hours after application, continuing to work through the night while you sleep. Together, they attack the same problem from two completely different biological angles simultaneously. The acid dissolves the cellular wall of the active fungal colonies. The alkaline residue destroys the pH environment required for any surviving spores to regrow. The fungus has no biological mechanism to survive both attacks occurring at the same time. No adaptation available. No resistance pathway. And the total cost of both ingredients combined is under $3 at any grocery store in America. Under $3. For a two-sided biological attack on the exact fungal mechanism that a $600 prescription bottle addresses from only one angle with a documented 17% success rate. Now here is where the numbers become genuinely uncomfortable to sit with. The global antifungal drug market was valued at $9.4 billion in 2024. That figure comes from verified pharmaceutical market research published in early 2025 following comprehensive industry analysis. The corporations inside that market include Novartis, Bayer, Bausch Health, Pfizer, and dozens of other household name pharmaceutical companies, all selling prescription and over-the-counter antifungal treatments at price points that have no relationship to their actual effectiveness. The most commonly prescribed oral treatment for toenail fungus costs between $350 and $900 for a full treatment course in the United States, depending on insurance coverage and pharmacy pricing. It requires 12 weeks of daily dosing. It requires liver function monitoring through blood work. and it achieves full clearance in fewer than 40% of patients, according to the treatment's own published clinical trial data. Their own numbers, fewer than 40%. Now look at the topical prescription alternative. A product called Jublia, one of the most heavily advertised prescription antifungal treatments currently on the American market, costs over $600 for a single bottle in the United States, without insurance coverage. Its own FDA-submitted clinical trials, the data the company itself submitted to receive approval, report a complete cure rate of 17.8%. 17.8% for $600. That means 82 out of every 100 people who purchase that product, follow the instructions, complete the full treatment course, will not achieve a complete cure. They will be back. They will need another prescription, another bottle, another $600, another follow-up visit. And that is not a failure of the product from the perspective of the business model. That is the business model working exactly as designed. You cannot patent sodium bicarbonate. It has been a common household compound for over 170 years. You cannot trademark the pH scale. You cannot build a quarterly earnings report around a box of baking soda that costs under $1, lasts for months, and is available at every grocery store on Earth. The entire revenue architecture of pharmaceutical dermatology depends on chronic treatment cycles, repeat prescriptions, follow-up appointments, monitoring visits, insurance billing at each stage. A permanent $1 solution that people can apply in their own home destroys every single layer of that revenue model at once. So instead of the truth, you were handed a $600 bottle with a 17.8% success rate and told it represented the best that modern medicine could offer you. It was not the best medicine they could offer. It was the best business they could build. And there is a difference between those two things that the industry has spent billions of dollars in advertising, making sure you never stopped long enough to notice. Subscribe and share this forward right now. Every person who watches this video is one fewer patient paying $600 for an 82% failure rate. One fewer person spending 12 weeks on a prescription that requires liver monitoring just to take safely. Share this with someone who has been trying to fix this for years and getting nowhere. They have already paid enough. Here is exactly what you do tonight. You need 1 box of baking soda and 1 bottle of raw, unfiltered apple cider vinegar. Both are available at any grocery store, any pharmacy, any supermarket in the country. Total combined cost under $3. You do not need a prescription. You do not need an appointment. You do not need insurance coverage for what you are about to do. Step 1. Fill a clean basin with comfortably warm water. Add 2 full tablespoons of raw, unfiltered apple cider vinegar directly to the water. Soak the infected foot for exactly 15 minutes. Do not rush this step. During those 15 minutes, the acetic acid is actively softening the thickened nail plate and beginning direct contact penetration toward the nail bed underneath. This is the preparation step that makes everything that follows significantly more effective than baking soda applied to a dry, unsoftened nail. Step 2. Remove your foot from the basin and dry it completely. Every part of it. Between every toe, under the nail edge, completely dry. This step is not optional, and it is not a formality. Moisture is the primary environmental condition that fungal organisms require to survive and spread. If you leave any moisture present before the next step, you are actively working against the treatment you are about to apply. Dry completely. Step 3. Mix 2 tablespoons of baking soda with just enough clean water to form a thick paste. The consistency should hold its shape. Not runny. Not crumbling. A paste that stays where you put it. Apply this paste directly and generously to the infected nail and the surrounding skin on all sides. Press it gently into the nail edge and beneath the front of the nail tip as far as is comfortable. Cover the treated nail with a clean cloth or a clean sock. Leave it in place for a minimum of 30 minutes. For maximum effectiveness, leave it overnight. During this time, the sodium bicarbonate is penetrating through the softened nail plate and shifting the pH environment of the nail bed itself. The alkaline residue is making direct contact with the dermatophyte colonies anchored in the keratin layer. The environment the fungus requires to survive is being chemically dismantled beneath the nail surface while you sit still. Step 4. Rinse the foot with clean water and dry it completely again. Apply no moisturizer to the treated nail or surrounding skin after this step on treatment nights. Repeat this complete protocol, all 4 steps, every single night for 3 consecutive nights. By the morning of day 3, you will see visible changes beginning. The discoloration will begin shifting. The nail texture will begin normalizing. The separation, if present, will begin closing. These are not cosmetic changes. These are the visible signs of active fungal colonies being destroyed and the nail bed environment shifting outside the pH range that dermatophytes can survive in. After the first 3 nights, continue the same protocol every other night for 2 additional weeks. This extended phase is not optional for permanent results. The 3-day intensive phase destroys the active surface colonies. The 2-week maintenance phase permanently eliminates surviving root structures deeper in the nail bed that the first 3 treatments have weakened but not yet fully reached. No fumes. No chemical odor. No liver monitoring. No follow-up blood work. No prescription renewal. No 600 bottle. Safe for every skin type. Safe for individuals with diabetes who are often told pharmaceutical antifungals carry additional risks for them specifically. Safe for elderly individuals who cannot tolerate the systemic side effects of oral antifungal medications. Safe for use around children and pets with no ventilation requirements. One complete treatment course. Permanent results. Total cost less than a cup of coffee. Your grandmother did not spend her life fighting toenail fungus because she had better luck than you. She had better information. Information that passed freely between families for generations at zero cost, through zero prescriptions, with zero side effects, and zero follow-up appointments that needed to be billed to an insurance company. A solution so simple and so available that it required nothing from any commercial supplier to access. Then 50 years of pharmaceutical advertising, shelf placement strategy, doctor education programs funded by drug manufacturers, and regulatory systems shaped by industry lobbying convinced an entire generation that the old ways were primitive, that natural compounds were unreliable, that only patented laboratory chemistry was powerful enough to protect them. And it worked. The market hit $9.4 billion. The 17.8% success rate got quietly buried inside footnote text on page 47 of a clinical submission document that no patient was ever expected to read. And millions of people kept paying $600 for a bottle that failed 82 out of every 100 of them every single time, and then went back and paid again because no one told them there was another way. Now you know, and knowing is the beginning of something they cannot patent, cannot bury, and cannot take back from you. Share this with someone whose nails have been changing for years. Someone who has tried the pharmacy options and gotten nowhere. Someone who has been told by their doctor that this is just something they have to manage and live with. They have already paid enough. Subscribe so you never miss what they are still trying to keep from reaching you. What is inside the next video is heavier than this one,